All right, so welcome back to AFF. I was trying to remember exactly where we left off last time, and I think it was talking a bit about fish gills and kind of options for how we will arrange a circulatory system around these gills. So let's put the water moving in this direction. Our respiratory membrane here, and then we have an option here, engineering-wise, of either having the circulatory system with its blood moving in this direction, in which case we've set up what's called a countercurrent system. Or the other option would be to move the water and the blood in the same direction across the respiratory membrane or gill, and this would be a concurrent system. With a concurrent system, think about the water and the blood moving in the same direction. In a countercurrent system, things are moving in the opposite direction across the respiratory membrane. Now, this is an interesting thought experiment because it turns out you don't tend to find these concurrent systems. So what we're going to do is lay out basically the problem with it and then show you why this countercurrent system is a much better solution to a problem and why we tend to find it in a lot of organisms, including fish. All right, in the concurrent system, let's imagine that the water moving across the gills has in it the maximum amount of oxygen that could possibly be dissolved in this water. Now, in truth, the amount of oxygen that could be there is related in part to pressure, temperature of the water, etc. But let's just assume that this water coming in has 100% of the oxygen that could be in that water at that particular point in time. That's saturated water, water saturated with 100% of the oxygen that could be there. And let's further assume that the blood that's arriving at the gills has been completely stripped clean of all the oxygen in the tissues. Okay? So, we have the water and the blood moving across a very thin respiratory membrane, so that should set up a situation where diffusion should occur fairly quickly because there's a large difference in the concentration of oxygen, or the partial pressure of oxygen, in these two locations. So as the oxygen diffuses, that should increase the amount of oxygen in the blood and decrease the amount of oxygen in the water. So as this water moves, it's going to begin to lose oxygen, and as the blood moves, the blood begins to gain oxygen. So you could imagine a situation here where we begin to decrease oxygen concentrations in the water and increase them in the blood, that we would still allow for diffusion to occur, but because the concentration difference isn't as large as what we started with, the amount of diffusion occurring is much less. Okay? And as our thought experiment continues, imagine that if we were able to move as much oxygen as possible, that we would arrive at a situation where the concentration of oxygen in the water and the blood are equal, and therefore there's no more net diffusion. So under a concurrent system, because of the engineering of the tissues, the circulatory system relative to the respiratory membrane, etc., we have a fatal flaw in the system, and that is you can't crank up oxygen concentrations higher than 50%. So in your figures, and if this is too small, I'd recommend that you pull up the PowerPoints from Blackboard and move along with them if that's easier for you. But in this concurrent flow system, again, 50%, 50%. Okay? Now, on the other hand, the countercurrent system is much more elegant here. If we start out with 0% oxygen in the blood and the maximum amount of oxygen that we can place in the water at this location, you can imagine that no matter what the concentration is of oxygen in the blood after it's passed across the respiratory membrane, if we get it to any concentration less than 100%, then oxygen is still going to diffuse. So let's imagine that we've done a very good job through this process of picking up oxygen. Diffusion is still going to occur at this situation. Right? We can still crank up oxygen concentrations in the blood to any value less than 100% of the saturation. As we move down here, as long as I have less than that concentration in the blood, I can still move the oxygen. So basically this countercurrent system is allowing for oxygen to diffuse across the respiratory membrane at all of these locations, whereas in the concurrent system, high diffusion, lower, 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 lower diffusion until it stops. Here, diffusion is allowed to occur across the entire respiratory surface. Now, in order to make this work properly, we do have to control the movement of water. We have to ensure that the water is always moving in the opposite direction as the blood. So to accomplish that, we're going to force the water to move through or across the gills in a very specific direction. Okay? Just another view here. There's the countercurrent system. Here are the blood vessels, the blood moving through the gills. And in order to force the water across the gills in a specific direction, we need to move the water in this direction. That is, in through the mouth and out through that opening on the side here that's covered by this plate called the operculum. So we'll say that water comes in through the mouth and out through the operculum. All right, if a fish is at rest, it needs to make sure that the water is always moving in the proper direction. So what we're going to do is set up basically a pumping series to channel the water through in a particular direction. So let's put the mouth here. Okay, that's the opening to the mouth. And then we're going to build this first cavity here. This is called the buccal cavity. Okay. All right, the second cavity here is where the gills are. This one is called the opercular cavity. This would be the opening out of the operculum where that little plate is. Right? So let's put that plate in place. Here, there would be another plate here, or the ability to close off the mouth at various points in time. Right? This, then, is the opening between the two cavities. So, let's close the mouth and pressurize this cavity, this buccal cavity. So we'll close the mouth and put this water under pressure. And as we do that, water would be forced into the opercular cavity. Okay? Now, to facilitate this process, we could squeeze on the water to positively pressurize the buccal cavity, or at the same time, I could either do it at the same time, or instead of pressurizing the buccal cavity, I could put the opercular cavity under negative pressure. The way to do that would be to open up this opercular cavity. So the muscles around this cavity relax and allow for expansion of that cavity. That increases the volume. That should have the effect of dropping the pressure. Pressure is higher now in the buccal cavity than it is in the opercular cavity, and water should move in. Okay? So here are pumps here, right? I can pull on that pump, decrease the pressure in the opercular cavity, pull the water in, okay? Now I'm going to start to pressurize the water in the buccal cavity to push even more water into the opercular cavity, right? So I've gone from a low to a higher pressure to a much higher pressure in the buccal cavity, continuing to force water in, and then I'm going to start to decrease the volume of the opercular cavity, but because I'm still pressurizing the buccal cavity, pressure is higher here than here, water should go out through the operculum. So what we have is this kind of chain reaction of pumps here. I've got this pump allowing for increased volume of the opercular cavity. It comes down. This one pushes up, keeps the water moving in. This one starts to push up, forces water out through the operculum. Decreased pressure pulls water in through the mouth. Then I start to drop the pressure in the opercular cavity as I increase the pressure in the buccal cavity. And you get this kind of series of pumps moving to maintain the flow of water unidirectionally across the gills. This control over the movement of water allows me to maintain this countercurrent system. Okay? All right. As with all the stuff we're doing, I know most of you are interested in humans. Keep in mind it's an animal form and function, not a human form and function. But also keep in mind that these very basic principles are important for all organisms. And if we can understand these kind of simple systems, it often makes understanding the more complex systems a little bit easier. Right? These problems and solutions that organisms face and come up with, and they aren't going away, right? These problems exist for everyone. All right, the next system to show you is the system that's used for respiratory surfaces in insects. Okay? All right, so imagine, if you will, a network of tubes running through this grasshopper, and this network of tubes has openings on the outside of the organism. So if this is my grasshopper here, right? Okay? Along the body of this grasshopper, there should be all of these tiny little holes. Okay? These tiny little holes are called spiracles. And these spiracles open up into tubes called trachea when they're large, or tracheoles when they're smaller. So here is that spiracle, large tube trachea, branching into smaller tubes called tracheoles. So there's that spiracle opening up into a larger tube, right? Get this picture correct here. That makes this the trachea, and there should be multiple of these, right, associated with each spiracle. And then we get to these smaller ones called tracheoles. Okay? These smaller tubes continue to get smaller and smaller, and they innervate individual cells. Right, so there are individual cells there. So with this network of tubes, you can imagine oxygen out here. CO2 is being produced in the cells because of the activity of cellular respiration in the mitochondria specifically, producing that carbon dioxide. And then what we have here is a system of tubes that allows for the diffusion of gases. If oxygen is going to get in there to those cells, it's going to have to diffuse down through these tubes and into the cells where the mitochondria are. The CO2 being produced here is at a higher concentration, and it should begin to diffuse out through this tracheal system. Okay? All right, here's this little flea here. This is a map of this kind of tracheal system as it moves through the insect. Now, a couple of things should occur to you. The first is, even though I have this network of tubes, the way I've presented it, the main force that's moving oxygen and CO2 around is diffusion. And if that's the force moving things around, it's super sensitive to differences in concentration as well as distances. We know that diffusion works better when there are differences in concentration that are much larger. Well, I can get that to occur. Lots of oxygen out here, not much near the mitochondria. Lots of CO2 near the mitochondria, not much CO2 out there in the real world. So I can do the difference in concentration. The distance problem is a bigger one. So as we think about kind of this tracheal system, the first thing we should conclude is, well, if I'm using diffusion, I'm talking about really short distances. And if you think about the size of your average flea, right, or your average aphid or any other kind of small insect like an ant, diffusion is going to be okay to work over these short distances. That make sense? Once the insect gets a little larger, this system as it's designed here is not going to work, because now I'm asking oxygen to diffuse over much longer distances, like we would find in a honeybee or even that grasshopper that I showed you. So for those insects, they have another solution on top of this network of tracheoles. And that's these structures here